Good afternoon to all. Yesterday we have discussed about the animal tissue culture, an introduction to animal tissue culture. Uh, Yesterday we discussed about yesterday we discussed about the introduction to animal tissue culture. Today we are here to discuss about the animal cell culture. Uh, what are the environment we have to provide for the good for the successful animal cell culture. Uh, what is the environment which influences on cell culture is the physiochemical aspects. Those were temperature, pH, osmotic pressure, oxygen, and CO2. As we discussed yesterday, we need a amount of 5% CO2 for the perfect growth. When coming to the physiological environment, hormones and the nutrient concentrations in which the cells propagate, while the physiological environment of the culture is not as well defined as its physico-chemical environment, a better understanding of components of serum, the identification of the growth factors necessary for the proliferation of the cells, and a better appreciation of the microenvironment of cells in the cultures. Here, the cell to cell interactions and the diffusion of gases and interaction with the matrix now allow the culture of the certain cell lines in the serum free medium. After all the considerations for creating an optimized cell culture environment for our cells, first we have to check the pH levels. Here the pH levels, most normal mammalian cell cultures grow well at the pH of 7.4, and there is a very little variability among different cell strains. However, some transformed cell lines have been shown to grow better at a slightly modified acidic environments. Acidic means 7 to 7.4, and some normal fibroblast cell lines prefer slightly more basic environments, that is 7.4 to 7.7. And insect cell lines such as SF9 and SF21 grow optimally at pH 6.2. How much CO2 levels we need for the perfect growth? The growth medium controls the pH of culture and buffers the cells in the culture against changes in pH. Usually, this buffering is achieved by including an organic or CO2 bicarbonate based buffer, because the pH of the medium is dependent on the delicate balance of dissolved carbon dioxide and bicarbonate, which changes the atmospheric CO2 can alter the pH of the medium. Therefore, it is necessary to use exogenous CO2 when using the media buffered with CO2 bicarbonate based buffer, especially if the cells are cultured in open dishes or transformed cell lines are cultured at high concentrations, while most researchers use mostly 5% to 7% CO2 in air. 4 to 10% carbon dioxide is normally or commonly for most cell culture environment experiments. However, each medium has a recommended CO2 tension and bicarbonate concentration to achieve the correct pH and osmolality. Uh, for HEK cell lines, we need 5%, and breast cancer cell lines also we need 5% of CO2. Optimal temperatures, usually optimal temperature is of 37 degrees centigrade. Uh, Optimal temperatures for various cell lines varies. The optimal temperatures for the cell culture mostly depending upon the body temperature of the host from which the cells were isolated, and to a lesser degree on the anatomical variations in the temperature. Those are temperature of the skin, and maybe lower than the temperature of the skeletal muscle. Overheating is more serious problem than underheating for the cell cultures. Therefore, the temperature in the incubator is slightly lower than the optimal temperature. So here we need 30 to 36 to 37 degrees centigrade for the optimal growth of human mammalian cell lines. And insect cell lines, we need 27 degrees centigrade for the optimal growth. And at 27 to 30 degrees centigrade. In between the 27 to 30 degrees centigrade, its growth is slowly lower, and more above 30 degrees, the availability of insect cells decreases, and the cells do not recover even after they are returned to the 27 degrees. Avian cell lines, which requires 38 degrees centigrade, 38.5 degrees centigrade, and cell lines which is derived from the cold-blooded animals like amphibians, cold water fish, which can tolerate a wide temperature range between 15 degrees centigrade to 26 degrees centigrade. What are the cell culture equipments we need? The specific requirements of the cell culture laboratory depending upon mainly the type of research conducted. For example, the needs of the mammalian cell culture laboratory, specializing in cancer research, is quite different from that of the insect cell culture laboratories that focus on protein expression. However, all cell culture laboratories have the common equipment or requirement of being free from pathogenic microorganisms bacteria, asepsis, and share some of the basic equipments that is essential for the culturing cells are the incubators, with incubator name itself indicating that incubator to provide the appropriate environment for cell growth. The incubator should be large enough for our laboratory needs, have forced air circulation, and should have temperature control to within plus or minus 0 0.2 degrees centigrade. A stainless steel incubators allow easy cleaning and provide corrosion protection, especially if humid air is required for incubation. Although the requirement for aseptic conditions in a cell culture incubator is not as stringent as that in a cell culture hood. Frequent cleaning of the incubator is essential to avoid the contamination of the cell culture. And what are the types of incubators? Here we have dry incubators and humid CO2 incubators. Dry incubators are more essential and economical, but requires the cell cultures to be incubated in sealed flasks to prevent evaporation. And but placing a water dish in a dry incubator can provide some humidity, but They do not allow precise control of atmospheric conditions in the incubator. Humid CO2 incubators are more expensive but allow superior control of culture conditions. They can be used to incubate cells cultured in petri dishes or multi-well plates, which require a controlled atmosphere of high humidity and increased CO2 tension. Storage. A cell culture laboratory should have store areas for the liquids such as media and reagents for chemicals. Those are drugs and antibiotics. For consumables such as disposable pipettes, culture vessels, and gloves. For glassware such as media bottles and glass pipettes. For specialized equipment. And for tissues and cells, glassware, plastic, and specialized equipment can be stored at ambient temperature on the shelves and in drawers. However, it is more important to store all media reagents and chemicals according to the instructions uh, which is there on the bottles, which is there on the bottles. And some media reagents and chemicals are sensitive to light, while other while normal laboratory use under light conditions is tolerated. They should be stored in the dark or wrapped in aluminum foil when not in used. So we need specifically for cell culture laboratories, we need a specific storage places for the all consumables and media, reagents, and the chemicals. Otherwise, there is a prone for the contamination. And also refrigerators, as just we discussed, we need a small cell culture laboratories. A domestic refrigerators is enough, and preferably one without auto defrost uh, refrigerator, which is like 2 to 8 degrees. Along with that, we need minus 80 also we need for the storage of cell lines after the passage. Such is like we need minus 5 degrees centigrade and minus 20 degrees centigrade and minus 80 degrees centigrade freezers we need for cryo storage. Because at different levels we will maintain the temperature for cryopreservation. At first, we will make the cells to be arrested in normal 4 degrees centigrade, and then we will transfer that to minus 20 degrees centigrade, and then finally we will keep the vial in the minus 80-degree freezer. So that we need the freezers of 4 degrees, refrigerators of 4 degrees, 2 to 8 degrees, and freezers of minus 20 degrees and minus 80 degrees. Uh, while most of the reagents can withstand the temperature oscillations in an auto defrost, like self-thawing freezer, some reagents such as antibiotics and enzymes should be stored in freezer that does not auto defrost. And as just we discussed, cryo storage. There are two types of liquid nitrogen storage systems. One is a vaporized phase and a liquid phase. Welcome as a wide-necked or narrow-necked storage containers. Vapor phase systems minimize the risk of explosion with cryo storage tubes and are required for storing biohazardous materials, while the liquid phase systems usually have longer static holding times and are therefore more economical. And the narrow-necked containers have a slower nitrogen evaporation rate, are more economical but wide-necked containers allow easier access, have a larger storage capacity. Here we need a cell counter. Definitely we need cell counter. Uh, after a cell counter is essential for quantitative growth kinetics and great advantage when more than two or three cell lines are cultured in the laboratory. Here the history of animal cell culture is started. Yesterday also we discussed about the history of animal culture. Started with Claude Bernard in 1878 proposed that physiological systems of an organism can be maintained in a living system after the death of organisms. And in 1885, rocks maintained embryonic chick cells in the saline culture. And 1897, Lom demonstrated the survival of cells isolated from the blood and connective tissue in serum and plasma. In 1903, Joly observed cell division of salamander leukocytes in vitro. In 1907, Harrison cultivated frog nerve cells in lymph clot held by the hanging drop method and observed the growth of nerve fibers in vitro for several weeks. He was considered by some of uh, he was considered some as the father of cell culture. Then the father of cell culture is Harrison. Burroughs succeeded in long-term cultivation of chicken embryo cell in plasma clots. He made detailed observations of mitosis in 1910. In 1911, Lewis and Lewis made the first liquid media consisting of seawater, serum, embryo extract, salts, and peptone. They observed limited monolayer growth. In the year of 1913, Carroll introduced a strict aseptic techniques so that cells could be cultured for longer periods. In 1916, Ross and Johns introduced proteolytic enzymes trypsin for the subculturing of adherent cells. Carroll and Baker, they developed a Carroll or T-shaped flask as the first specifically designed cell culture vessel. They employed microscopic evaluation of cells in culture. In 1927, Carroll and Rivera produced the first viral vaccine, Vaccinia. In 1913, Gay developed the roller tube technique. In 1940, the use of antibiotic penicillin and streptomycin in culture medium to decrease the problem of sorry decrease the problem of contamination in the cell culture. In 1948, Earle isolated mouse L fibroblast, which formed clones from single cells. Fisher developed a chemically defined media, CMRL 1066. In 1949, Enders reported that poliovirus could be grown on human embryonic cells in culture. In 1952, Gade established a continuous cell line from a human cervical carcinoma known as Helen Lane cells, human cervical carcinoma cells. Dalbecco developed plague assay for animal viruses using confluent monolayers of cultured cells. Right now we have Dalbec media for cells. In 1954, Abercrombie observed contract inhibition motility of diploid cells in monolayer cultures ceases when contact is made with adjacent cells. In 1955, Eagle studied the nutrient requirements of selected cells in culture and established the first widely used chemical defined medium. Even now also people are using eagle nutrient requirement medium for cell culture. In 1961, Hayflick Moorhead isolated human fibroblast WI38 and showed that they have a finite lifespan in cell culture. Littlefield introduced the hat medium for cell selection in 1964. In 1965, Ham introduced the first serum-free medium which was able to support the growth of some cells. In the same year, 1965, Harrison and Hatkins were able to fuse human and mouse cells by the use of a virus. In 1975, after 10 years, Kohler and Milstein produced the first hybridoma capable of secreting monoclonal antibody. We already discussed this point in the introduction of biotechnology. Kohler and Milstein produced the first hybridoma capable of secreting monoclonal antibody. In 1978, Sato established the basis for the development of serum-free media from cocktails of the hormones and growth factors. In 1982, human insulin became the first recombinant protein to be licensed as a therapeutic agent. In 1985, human growth hormones produced from recombinant bacteria was accepted for therapeutic uses. In 1986, lymphoblastoid Gamma IFN licensed. In 1987, tissue type plasminogen activator TPA from recombinant animal cells became commercially available. In 1989, recombinant erythroprotein in trial. In 1990, recombinant products in clinical trial, HBS AG factor 8, HIV GP120, HIV glycoprotein 120, and CD4 markers, and GM, CSF, and EGF, and monoclonal antibodies, and interleukins too. All these are in the clinical trials. What are the basic tissue culture techniques? We will see tissue culture is the method of biological research in which fragments of tissue from an animal or plant are transferred to an artificial environment for the survival and the function. The cultured tissue may consisting of a single cell, a population of cells, or a whole or part of an organism. Cells in culture may multiply, change size, form, or function, exhibit specialized activity. Muscle cells, for example, may contract, interact with other cells. Suspension cultures. Suspension cultures are generally used for anchorage dependent and anchorage independent. What are and anchorage independent cells? Yesterday also we discussed is the same point, such as hemopoietic cells, some transferred cell lines, and for free floating cells or cells formation. Those are isolated cells, aggregates, or tissue fragments. And anchorage independent cells are grown in either a semi-solid medium, such as agarose, or in fluid suspension culture under continuous agitation, stirring, and shaking. Short-term suspension cultures of bulk isolated brain cells have also been described by Verity in 1995. In general, suspension cultures are easy to maintain and are ideal for scale-up. For the latter, culture vessels of various sizes are available, ranging from small spinner flasks to industrial size fermenters, which is explained by Jacobi and Pastan in 1979. What is monolayer? That is attached monolayer. A monolayer cell culture is the most wide usually technique which numerous protocol variants of this approach practiced in different laboratories in these cells and in these cultures. The cells grow attached to the surface of the culture vessel, although specific treatment of the culture vessel surface with polylysin and polyornithine may be necessary for betterment, cell supplement, and the growth conditions are also influenced by the cell density techniques, which different cell density techniques ranging from single cell microcultures to high density Mars cultures. The yield in mono layer cultures is limited by the available surface to which the cells may attach. Therefore, for scaling up, cells may be grown in roller bottles, that is cylindrical bottles, and here we need to continuously supply the medium partially filled. The cylindrical bottles are first partially filled with the medium and rotated around their horizontal axis at 40 to 50 rpm to provide continuous medium supply. In capillary perfusion systems, in which medium is circulated through a tightly packed artificial capillaries or attached to the particulate microcarriers that can be maintained in suspension cultures. With a high cell density culture, essential metabolites may be rapidly depleted, causing density dependent growth inhibition and eventually cell death. One advantage of monolayer cultures is their ready accessibility for direct microscopic examinations as well as for morphological, immunocytochemical, and electrophysical physiological studies. On the other hand, it is relatively difficult to sample monolayer cultures for ultrastructural or biochemical analysis without destroying the culture. Furthermore, with these cultures, the cells are attached to an artificial substrate rather than to the natural extracellular matrix, limiting directed cell to cell interactions. There are three-dimensional aggregated cell cultures. Aggregated cultures are prepared from disassociated cells allowed to re-aggregated under the controlled conditions and continuous gradatory agitation to form regular spherical cell structures, such as cultures permitted a maximum of cell-to-cell interactions and thus development of natural cell matrix and histotypic cell formations. Aggregated cell cultures prepared from the fetal cells show a particular high capacity for cellular organization and maturation. Although it is been shown by the scientist Moscana, who invented this particular cell culture technique in the year of 1965, that immature cells of any tissue are able to re-aggregate and mature into histotypic structures. This method is most suitable for immature neural cells. One advantage of aggregated cell cultures is that they provide a large number of highly reproducible replicates from which aliquots are already sampled for multidisciplinary studies. On the other hand, aggregated cultures do not permit the direct microscopic observations of the cells during the cell culture. Furthermore, three-dimensional cultures, oxygen and nutritional gradients are critical and limiting the size of the individual aggregate to a diameter less than 400 micrometer. Moreover, in certain preparations such as aggregate cultures of fetal liver cells, an additional diffusion barrier is formed by a peripheral epithelioidal cell layer. This is not the problem which encountered with the aggregate cell cultures of fetal brain cells, which can be maintained for months at a highly differentiated stage. Tumor cells or explants grown in sponge gel matrices form three-dimensional structures Those are called as spheroids and exhibit tissue-specific drug responses. What is explant culture? Explant cultures are prepared either from intact organs such as a dorsal root ganglia and sympathetic ganglia and parasympathetic ciliary ganglia, or from small tissue fragments that do not exceed 1 millimeter in any dimension. This approach is particularly useful for neural tissues, which, due to their structural and functional complexity, are more difficult to reconstitute in vitro. Explants may be maintained in stationary culture or cover glasses or cover slips. There may be replaced on porous, other may be placed on porous and transparent membranes that remain in contact with the liquid medium in such a way that a thin film of liquid surrounds the tissue. Often the attachment of the explants is enhanced by substrate coating using collagen, laminin, or polylysin as a further variant. Brain slices embedded in a plasma clot on glass cover slip are maintained in a roller tube, which allows for altering exposure of tissue to air and liquid medium. Explant or micro explants from minced tissue may also be maintained in non-adherent organ culture by using continuous gyratory agitation to keep the tissue in suspension. A stationary as well as a free-floating explants preserve their cytoarchitecture and gross anatomic cellular organization to a large extent, although structural and functional abnormalities related to their in vivo situation occur. Roller tube slice cultures decrease in thickness to 1 to 3 cell layers within a few days of culture, but generally retain their histotypic organization. This preparation is particularly well suited for direct microscopic observation and direct microscopic observation and electrophysical studies. As a rule, explant cultures are the best approximation of in vivo situations with respect to the organ specific cellular organization. For example, cerebral explants develop normal architectural arrangements of cortical laminae and deep nuclei, and hippocampal explants from functional sympathetic networks in culture, as well as normal dendritic arborizations of pyramidal and granular cells. Explant cultures are also offer a normal dendritic arborizations of pyramidal granular cells, and it is a unique system for the study of adjacent brain regions in co-cultures, such as they are able to form the interacting afferent and efferent projections under the in vitro condition. The only main disadvantage of expand culture is the smaller amount of the tissue available and relatively low number and limited reproducibility of replicate cultures, making it difficult to use them for biochemical and molecular biology studies. So what is the media here is natural media. Here we have two types of media. One is artificial media and natural media. Natural media, which includes the naturally occurring biological fluids, that is are of three types. One is clots uh, and biological fluids and tissue extracts. Clots, plasma clots are the most commonly used clots that have been employed for a longer time. In the present time, plasma is commercially found in liquid state. that can be prepared in the laboratory. And biological fluids, like several biological fluids can be employed as a culture media, such as amniotic fluid and pleural and ascitic fluids and hemolymph of insects, aqueous humoral from eye, serum. Among them, serum is the most preferred one. And when coming to the tissue extracts, the most commonly used tissue extract is chick embryo extract. However, bovine embryo extract is also used in the culture media. The extracts from the spleen, liver, bone marrow, and leukocytes were also used. The substitution for the tissue extract can be mixture of the amino acids and some other organic compounds. When coming to the artificial media, for the purpose of various artificial media have been employed for immediate survival, a balanced salt solution with specified pH and adequate osmotic pressure, and prolonged survival, a balanced salt solution in addition with serum or appropriate formulations of organic compounds, indefinite growth and specified functions. Artificial media may be classified into four types. One is serum containing media and serum free media, protein free media, and chemically defined media. And what is serum containing medium? Here, serum containing media is the yellowish liquid medium, which is a transparent content that remains left over after the removal of fibrin and cells from the blood. 2 to 10% of serum is often contained by normal growth media. The most commonly employed supplement in animal cell culture is fetal bovine serum, FBS. It supplies the basic nutrients for the cells. It also contains several growth hormones and various growth factors. In addition to this, it is also containing the buffer. The serum containing media in cell culture media, um, mm, which carries water insoluble nutrients and protease inhibitors, growth hormones, and growth factors, and binding and neutralizing the toxic moieties. It is employed as a economically supplement media. And serum free media. Right now, we talked about serum containing media and serum free media. In case of immunological studies, if we want to study any immunological studies, the presence of serum in the media can give the misinterpretations, like a very serious misinterpretations. That's what, in generally, these serum-free media are specifically designed to promote the culture of a single type of cell and incorporate a specified amount of purified growth factors, lipoproteins, and other proteins normally supplied by the serum. As the components of these media are known, thus it is referred to as a defined culture media. And when coming to the chemically defined media, here the chemically defined media is may also contain pure protein additives and growth factors and vitamins, cholesterol, and these constituents are produced by genetic modification in bacteria or yeast, particular amino acids and fatty acids. And protein free media is devoid of any protein and only include non-protein constituents. Usage of protein free media enhances the superior cell growth and protein expression in contrast to serum supplemented media, and enables downstream purification of any expressed product. In order to choose a medium, the minimum criteria required usually involve the medium should supply The cells with all of nutrients keep the physiological pH about 7 with ample buffering. The medium should be sterile and isotonic to the cells. The balanced salt solution was initially established uh, a physiological pH and osmolarity needed to sustain the cells in vitro was the basis for any cell culture media. Various components like glucose, amino acids, vitamins, growth factors, antibiotics, etc., have been added to promote cell growth and proliferation, and several media have been created. When coming to the primary cell culture, primary versus secondary cell culture and their maintenance and preservation. As we discussed, primary cell culture, primary cells are directly straight away obtained from the host cell, and these cells are dissociated from the parental tissue or grown on a suitable container, and the culture thus obtained is called as primary cell culture. Such culture comprises mostly heterogeneous cells, and most of the cells divide only for a limited time. However, these cells are much similar to their parents, and depending upon the origin, primary cells grow either as an adherent monolayer or in a suspension culture. Adherent cells, these cells are anchorage dependent cells and propagate as a monolayer. These cells need to be attached to a solid or semi-solid substrate for proliferation. These adhered cells adhere to the vessel with the use of an extracellular matrix, which is generally derived from the tissues of organs that are immobile and embedded in a network of connective tissue. And fibroblast and epithelial cells are of such type of the adherent cells. When the bottom of the culture vessel is covered with continuous layer of cells, usually one cell thickness, these are known as monolayer cells. Majority of continuous cell lines grow as monolayers. As being single layers, such cells can be transferred directly to the cover slip to examine under the microscope. What are suspension cells? Suspension cells do not attach to the surface of the culture vessel. These cells are also called as anchorage independent or non-adherent cells, which can be grown floating in the culture medium. Hemopoietic stem cells, which is derived from the blood, spleen, and bone marrow, and tumor cells can be grown in suspensions. These cells grow much faster, which do not require the frequent replacement of the medium and can be easily maintained. These are of homogeneous cells, and enzyme treatment is do not required for dissociation of cells. Similarly, these cultures have short lag period where it can use more nutrients from the culture medium. When coming to the confluent culture and the necessary of the subculture, after the cells are isolated from the tissue and proliferated under the appropriate conditions, they occupy all the available substrate, which reaches to the stage called as confluence. For a few days, it can become too crowded for their container, and this can be detrimental. This can go to the detrimental to their growth, generally lead to the cell death if left for a longer period. Mostly cells go detrimental stage after 7 days. The cells thus have to be subculture A portion of cell is transferred to a new vessel with a fresh growth medium, which provides more space and nutrients for the continual growth of both portions of cells. Hence, subculture keeps cells healthy and in a growing stage. A passage number refers specifically to how many times a cell line has been subcultured, in contrast with the population doubling level, in that the specific number of the cells involved is not relevant. It simply gives a general indication of how old the cells may be for various assays. And secondary cell culture and cell line. When a primary culture is subcultured, it is known as secondary cell culture or cell line or subclone. The process involves removing the growth media, dissociate the adhered cells using enzymatically. Here we use a trypsin for the detachment of the cells. Subculturing of primary cells to different divisions leads to the generation of cell lines during the passage. Cells with the highest growth capacity is predominant, resulted in the degree of genotype and phenotype uniformity in the population. However, they subcultured serially, they become different from the original cell. Gene can be altered. On the basis of the lifespan of the culture, the cell lines are categorized into two types. One is finite cell lines and continuous cell lines. Finite cell lines go through a limited number of cell divisions having a limited lifespan, on the cells passage several times, and then those their ability to proliferate, which is a genetically determined event known as senescence. And cell lines derived from the primary cultures of normal cells are finite cell lines. Continuous cell lines, when a finite cell line undergo transformation and require the ability to divide independently, indefinitely, it becomes a continuous cell line. Such transformation or mutation can occur spontaneously or can be chemically or virally induced, or from the establishment of the cell cultures from malignant tissue. Cell cultures prepared in this way can be subcultured and grown indefinitely as a permanent cell lines and are immortal. These cells are less adhere, fast growing, and less fastidious in nutritional requirements, that can able to grow up higher cell density and different in phenotypes from the original tissue, such as grow more in suspension. They also have a tendency to grow on the top of each other in the multi-layers on culture vessel surfaces. Here you can see the animal cell culture. Here the cells are isolated by the enzymatic and mechanical action from the organ that can be cultured in the culture disc. Uh, here, uh, Here the production of number of cells, that is called as confluency or confluence and confluent culture, that can be subcultured. Here, when the subculture happened, it is known as secondary culture from the primary cell line, and the next secondary cell line. And when the passages happens, cell division continues up to finite divisions. And the other hand, uh, the when the organ is having a tumor, cells isolated from the tumor and cultured and transformed cells, when it is subcultured, cell division continues up to the indefinite division. This is called as cell line. This is the major difference between the cancer cell lines and normal cells. that is primary culture and the cancer cell line. This is the entire protocol, the entire picture gives us the information about the animal cell culture. This is the normal organ and this is the tumor organ which is having the tumor. When coming to the, even yesterday also we discussed about the primary cell culture and secondary cell culture. Again, here I want to give an idea about the primary cell culture and secondary cell culture. Here the primary cell culture is the growing and maintaining of the selected cell types exercised from a normal parental tissue. And when coming to the secondary cell culture, is a cell line or subclone cultured from the primary cell culture. This primary cell culture contains the cells directly obtained from the host tissue, either through mechanical or enzymatic digestion. In secondary cell culture, which contains the subcultured cells from the primary cell culture, a primary cell culture is a heterogeneous, whereas secondary cell culture is a homogeneous because it contains only one type of cells. But primary cell culture contains different types of cells. That's what we called as heterogeneous. I have the same biological response as the cells in the host tissue. In case of secondary cells, which is adapted to the culture conditions by altering their biological mechanisms. Primary cell have similar genetic makeup to the cells of the host tissue, and secondary cells have altered genetic makeup. A primary cell do not undergo cell proliferation, but the secondary cell undergo cell proliferation. A primary cell do not contain a sufficient amount of cells, but the secondary cell have the optimal cell density. A primary cell have a finite lifespan, whereas secondary cell line have indefinite lifespan. A primary cell unable to maintain by passaging, but we can maintain the secondary cell line by passaging. A primary cell line requires a rich mixture of amino acids, micronutrients, certain hormones, and growth factors. But secondary cell lines, it is very easy to maintain. High risk, primary cell lines have the high risk of contamination. When we maintain the laboratory conditions, the secondary cell line growth is low risk of contamination. And primary cell culture serves as an in vivo model, whereas secondary cell line serves as an in vitro model. Primary cell lines important in manufacturing vaccines and therapeutic development, whereas the secondary cell lines important for the production of growth hormones, antibodies, and anti-cancer agents. This is about a clear picture of a primary cell culture and secondary cell culture. As we already discussed, culture maintenance. Today, a wide variety of ways are available to cell culture, incubators, vessels, and antibiotics, and that allow tightly conditions for culturing to avoid the contaminations and infections. But sometimes we will require to maintain the culture for a longer time. For this, the best method is cryopreservation and freezing of culture by using a cryoprotectant called as DMSO. DMSO is a dimethyl sulfoxide, but this is a very toxic agent. In these cells can be processed, cells preserved at very low temperatures, and on this cell tolerate a very low metabolic activity. What are the equipments we require for this one? Cultural maintenance is laminar airflow, and mechanical pipetter, inverted microscope, and vacuum pump, and flask, and CO2 incubator. What is the process? Here the cell culture protocol. Use only healthy cells or tissues. Detach the cells with the help of trypsin and then resuspend that cells into the growth medium. Adjust concentrations about 4 to 8 into 106 cells per millimeter per ml, and this can be um, by with the help of um, cell counter. We can count for the concentration, then fill to the culture in specific vials or ampoules, then freeze them slowly and then transfer to the vials. A gaseous phase mean or it liquid nitrogen with liquid nitrogen with minus 196 degree centigrade. How to recover that preserved cells, recovery of the preserved cells. In this process, preserved ampoules transfer on their water bath at 36 to 37 degrees centigrade, then transfer to the cells on the appropriate medium and allow to produce cell monolayer. In between, we have a simple process called like centrifugation for removing of that DMSO and other chemicals which is presented in the preservation of cells. And what are the applications of animal cell culture? Vaccine production, one of the most appropriated uses of cell culture in research and production of vaccines. The ability to grow the large amount of viruses in the cell culture eventually led to the creation of polio vaccine. And cells are still used today on a large scale to produce vaccines for many other diseases like rabies, chickenpox, hepatitis B, and measles. In early times, researchers had to use live animals to grow polio virus, but due to the development of cell culture techniques, they were able to achieve much greater control over virus production and on a much larger scale, which eventually developed vaccines and various treatments. However, continuous cell lines are not used in virus production for human viruses, as these are derived from the malignant tissue or possess malignant characteristics. Virus cultivation and study. Cell culture is widely used for the propagation of the viruses. It is convenient, economic, easy to handle, and compared to other animals, it is easy to observe cytopathic effects and easy to select particular cells on which the virus grow, as well as to study the infectious cycle. Cell lines are convenient for virus research because cell material is continuously available, and continuous cell lines have been extremely useful in the cultivating many viruses previously difficult or impossible to grow. And cellular and molecular biology. In cellular and molecular biology, cell culture is the one of the major tools used in this providing excellent model system for studying the normal physiology, biochemistry of the cells, that is metabolic studies and aging, the effects of different toxic compounds on the cells, and the mutagenesis and carcinogenesis. The major advantage of using cell culture for any of these applications is the consistency and reproducibility of results that can be obtained from using a batch of clonal cells. In cancer research, our normal cells can be transformed into cancer cells by methods including radiation, chemicals, and viruses. These cells can then be used to study cancer more closely and to test potential new treatments and drug developments. A gene therapy cells have a functional gene can be replaced to cells which are having non-functional gene and for which the cell culture technique is used. And immunological techniques, immunological studies, cell culture techniques are also used to know the working of various immune cells, cytokinins, lymphoid cells, and an interaction between the disease causing agents and the host cells. What are other techniques? Cell lines are also used in vitro fertilization technology, IVF, and recombinant protein and drug selection and improvement. Cell growth and growth kinetics of the animal cells. One second. Ah. Here, for any a cell culture like animal or plant cell culture, we need to refer the cell increase in size and cell divisions that have to be described the division of a mother cell into daughter cells, that from one cell to two, two to four, four to eight. The cell proliferation is the process in which animals that grow in the growth of the animals is more restricted in time, that is that of um, plants, but cell divisions is more generally distributed through the body of the organism, although the rate of cell division differs in different regions. But in case of um, embryo generating, an increased number of cells through the cell division, the capacity for the cell division is widely distributed in this developing embryo. In plants, plants are differ from the animals in the manner of growth. As a young animal mature, all parts of their bodies grow until they reach to a genetically determined size for each species. But in case of plant growth, a continuous growth throughout the lifespan of the plant can be seen is restricted to certain meristematic tissue regions only. When coming to the growth kinetics is an autocatalytic reaction which implies that the rate of growth is directly proportional to the concentration of the cell. The cell concentration is measured by direct and indirect methods. In the direct method, that includes measuring the cell mass concentration and cell number density by its dry weight, turbidity, and plate counts. That is optical density value and plate counts. Whereas in indirect methods of measuring cell density are done by measuring the concentrations of the proteins, ATP, or DNA content. And batch growth kinetics of a micro follows a growth curve with the lag phase as an initial phase during which cells adapt to the new environment. Multiple lag phases occur if the media is supplemented with more than one sugar, and such type of growth is referred as a diaxic growth. Following the lag phase is the log phase, in which here you can see the log phase, lag phase, and in the log phase in which the cell mass and cell number increase exponentially, then the depletion of the nutrients starts, in which indicates the deceleration of phase. The accumulation of the toxic products results in the deceleration of phase, after which stationary phase commences, in which growth rate equals the death rate. The continuous growth kinetics assessed by the perpetual feeding process in which the growth is controlled by the concentration of the rate limiting factors. Here you can see when the single inoculum or inoculation in the liquid media does not start dividing as per its generation time, during which the phase bacteria gets adjusted to the new media growth, not only the bacteria but the cells divide into the daughter cells. In phase bacteria synthesize the most crucial enzymes, coenzymes required for the optimal growth and multiplication. In addition, cell is metabolically active, be busy with the synthesizing large amount of the protoplasm. At the end of this phase, each bacterial cell divides and redivides and entering into the next phase of active multiplication. Here you can see the time. This is the typical growth curve for the batch system. Here you can see the lag phase. It is the first phase and acceleration phase where the nutrients is uptaken by the bacteria taken place. This is also called as a growth phase, a log phase. Here you can see the more viable cells. And once a cell is reached to a started taking the toxic substances presented in the media, then the decline phase starts. That is also called as a stationary phase, where the cells is stopped to taking the media and the requirements, and from there death of the cells are taken place. Then the last stage is death phase. Death phase, when the substrate is not sufficient from the dying cells, death rate of the bacteria superseded of growth, and as a result of bacteria declines sharply. Here we have a mathematical model, that is monad equation. It is named for the scientist Jacques Monod, a French biochemist. He received a Nobel Prize in Physiology or Medicine in 1965, who the one who proposed using a equation of this form to relate microbial growth rates in an aqueous environment to the concentration of the limiting nutrients. The Monod equation has the same form as a Michaelis and Menton's equation, but differ in that it is empirical while the latter is based on theoretical conditions. Here you can see the monad equation. Mu is equals to mu max. Here the mu is growth rate of the considering microorganism, whether it is the bacteria or cell, anything. So mu is the growth rate of the considered microorganism. Mu is equal to mu max. Here the mu max stands for the maximum growth rate of the organism. S is the concentration of the limiting substrate for the growth by Ks. Ks is the half velocity constant. The value of S when mu by mu max is equals to 0.5. Here you can see the monad coefficients to the monad equation. They will differ between the microorganism species and will also dependent on the ambient environmental conditions. For example, on the temperature, pH of the solution, on the composition of the culture medium. Here you can see the growth rate mu of a considered microorganisms as a function of the limiting factors. So there are various types of culture. cultures, microcarriers attached to growth suspension cultures, and continuous flow cultures. We already discussed about the monoculture and the rocks bottles. Here you can see the rocks bottle and the roller bottle. As we discussed rocks bottle, uh, um, generally we have cell culture used for obtaining useful products like biochemicals, interferons, interleukins, and hormones, enzymes, antibodies. The scaling of cell cultures, we need monoculture, suspension culture, and immobilized cultures. In case of monocultures, monolayer cultures, for scaling up, usually monolayer cultures are the essential for anchorage dependent cells. For scaling up of such cultures is based on their increase the available surface area by using the plates, spirals, ceramics, and microcarriers, which is most effective. The various culture vessels used are one rocks bottle, a roller bottle, and hollow fiber cartridge, and stack plate, and airlift fermentation. These five types of uh, various types of culture vessels which is used for scaling up of the animal cell cultures. First, coming to the rocks bottles, it is commonly used in the laboratory and it is kept stationary so that only a portion of its internal surface is available for the cell anchorage. Each bottle provides about 175 to 500 200 centimeter square cell surface area per cell attachment and occupies 750 to 1000 centimeters cube space. Here you can see the structure of roller flask, rocks flask. It looks like a T. And a roller bottle. This vessel permits a limited space scale up and it is rocked or preferably rolled so that its entire internal surface is available for anchoraging. Several modifications of the roller bottle further enhanced the availability. One is Spiracell, a spiral polystyrene cartridge, and glass tube, a roller bottle packed with a parallel cluster of small glass tubes separated by silicone spacer rings and extended surface area roller bottle. The bottle surface is corrugated and enhancing the surface by factor of 2. And the next one is a multi-tray unit. Here you can see the multi-tray stack plate. A multi-tray unit, a standard unit which contains 10 chamber stacks on each other, which have interconnected channels. This enables the various operations to be carried out in one go for all the chambers. Each chamber has a surface area of 600 centimeters square and the total volume of the units in 12.51, and this polystyrene unit is disposable and gives good results similar to the plastic flask. And synthetic hollow fiber cartridge, here you can see hollow fiber cartridge. The fibers enclosed in a sealing cartridge provides a large surface area for cell attachment on the outside surface of fibers. The capillary fibers, which is made up of acrylic polymer, which has the 350 micrometer in diameter with 75 micrometer thick walls. The medium is pumped into through the fiber. It perfuses through the fiber walls and becomes available to the cells. The surface area available is very high, that is up to 30 centimeters square by ml of medium volume. The system is mainly useful for suspension cells, but it is also suitable for cell anchorage if a polysulfone type fiber is used. What is the opticell culture system? It has the cylindrical ceramic cartridge, which is with 1 millimeter square channels, which runs through the length of the unit, and a perfusion loop to a reservoir is provided for environmental control, that is medium and gas. It gives about 40 centimeter square surface area per ml of the medium. It is suitable for the virus, cell surface antigen, and monoclonal antibody production, and for both suspension and monolayer cell cultures. And plastic film, which is made up of by the Teflon. Teflon is the fluoroethylene propylene copolymer, which is a biologically inert and highly permeable to the gas. Teflon bags, which is of 5 to 30 centimeters length and width, which filled with the cells, and medium 2 to 10 mm deeper, and serves as a good culture vessels. Cells attached to the inside surface of the bags. Alternatively, Teflon tubes are wrapped around a reel with a spacer, and the medium is pumped through the tube. Cells grow on the inside surface of the tube. A culture vessel called stericell is available. Helicell vessels. These vessels are packed with polystyrene rubbers that are twisted in helical shape. The medium is pumped through the vessel. The helical shape of the rubber ribbons ensuring good circulation. The cells adhere to the ribbon surface. All the culture vessels, in addition to the increased surface area due to the vessel design, allow further scaling up by the use of multiple units of the vessels. In contrast, the following three culture systems, which is used for the scaling up of the single unit by increasing the vessel volume. In addition, they make the monolayer culture system considerably similar to the suspension cultures. One is bead bed reactors, which provides the surface for the cell attachment, and heterogeneous reactor, which which contain the circular glass or stainless steel plates stacked with 5 to 7 apart and fitted to the central shaft, which provides an airlift pump is used for the mixing, or the shaft is rotated either vertically or horizontally. But there is a disadvantage of this heterogeneous reactor is the system is very low ratio of surface area to medium volume. That is the major chief disadvantage. And microcarrier cultures, these systems is used 90 to 300 pm diaparticles as a substrate for the cell attachment. Initially, dextran beads, Cephaldax A50, were used by the Vander Welch in 1967. They were not entirely satisfactory due to the unsuitable charge of the beads and possibly due to the toxic effects. The microcarriers available for using the range of present range for from the dextran a polystyrene and a polyacrylene, a glass, polyacrylamide, silica, DAE, a Sapodax media, and cellulose and gelatin to the collagen. The specific gravity of the microcarriers ranges from 1.02 to 1.05. A microcarriers greatly increase the surface area for cell attachment for unit medium volume, which can be up to 90,000 centimeters square per liter, depending on the size and density of the beads. Yes, there is a alternatively, beads may be dissolved where possible. Gelatinous beads are dissolved by the trypsin and collagen. Coated beads are treated with collagenase and dextrinase is used for the dextran bands, beads. These treatments leave the cells free which are collected. When coming to the next culture, suspension culture, as we know, suspension culture means scaling. This is the best condition for the scaling up. Both the chemical and physical factors have been optimized for the good result. The medium must be suitably stirred to keep the cells in suspension to make the culture homogeneous. It becomes increasingly difficult with the scaling up. Various types of stirrers range from simple magnetic stirrer and flat blade turbine impellers to marine impellers to those using pneumatic energy, for example, airlift fermenter, and those using hydraulic energy, a medium perfusion. Improved mixing can be obtained by changing the design of a stirred pedal or by using the multiple impellers. The objective of stirring is to achieve a good mixing without causing damage to the cells. A Vibro mixture achieves stirring by vertical reciprocating motion of zero point 0.1 SMM at a frequency of 50 cycles seconds of a mixing disc fixed horizontally to agitator shaft. These stirrers can cause a random mixing, less foaming, and lower shear forces. It is important to supply sufficient oxygen without damaging the cells. Main to utilization rate by cells in about 6 mg oxygen per 106 cells hour. But oxygen is only sparingly soluble in culture medium. The oxygen transfer rate from gas phase into the medium is about 17 micrograms per centimeters per hour. Therefore, surface aeration can support about 50 into 106 cells in 1 liter culture vessel when the medium depth is about 5 mm, especially above 5 centimeters. Aeration with a mixture of CO2 and air becomes necessary to maintain the adequate gas exchange. So the efficient and aeration is achieved by bubbling air through the medium, that is sparenging. But this may damage the animal cells due to the high surface energy of the bubble and on the cell membrane. The damage can be reduced by using the larger bubbles, lower gassing rates, and by adding the non-nutritional supplements like pluronic F6B polyglycol and sodium carboxymethyl cellulose and polyvinyl pyrrolidone, which protects cells from damage due to shear forces and bubbles respectively. Silicone tubing, high gas permeable, can be arranged inside the culture vessel 2 to 5 centimeters tubing of 30 meters length for a 1,000-liter culture, and air is passed through the tube. However, it is inconvenient to use. Aeration may be achieved by medium perfusion, in which medium is continuously taken from the cell culture, passed through an oxygenation chamber, and returned to the culture. The cells are removed from the medium taken for perfusion so that the medium can be suitably altered for pH control. Perfusion is used with the glass bead and more particularly with microcarrier system. When aware considered a safe and desirable, oxygen supply in the culture vessel can be enhanced with the normal 21% of higher value. Air pressure can be increased by 1 atmosphere. This increases O2 solubility and diffuses rate is, but there is a risk of oxygen toxicity. The reactors used for the large scale of suspension cultures are of three main types. One is stirred tank buffers and continuous flow reactors and airlift fermenters. So here the stirred tank bioreactors and airlift bioreactors. This we will cover for the next class. Then we'll go to immobilized cultures. And yes, so the major difference between these stirred tank bioreactors, these stirred tank bioreactors has the smaller vessels and stainless steel large volumes, and these are closed systems with fixed volumes are generally agitated with motor driven stirrers with considerable variation in the design, water jacket in place of heater type, temperature control, curved bottom for better mixing a low speeds, and mirror internal finishes to reduce the cell damage. Many heteroploid cell lines can be grown in such vessels. The needs for research biochemical uh, from cells are met from 2 to 50 on bioreactors, while large scale reactors are mainly used for growing hybridoma cells for the production of monoclonal antibodies through their yields from cultured cells. 1 to 2% of those obtained by passaging the cells through peritoneal cavity of the mice. And continuous flow cultures. The continuous flow cultures is also known as chemostatic and turbidostat type. In both the type and cultures begin as a batch culture. In chemostat type, inoculated cells grow to the maximum density where some nutrients, vitamins become growth limiting factor. A fresh medium is added after the 2 to 48 hours of the growth. A constant rate, usually um, lower than the maximum growth rate of culture, and at the equal rate the culture is drawn.